आप सबका नेट एग्जामिनेशन बहुत अच्छा हुआ होगा एज ऑलवेज. This time paper was comparatively average. We cannot say that it was very easy, nor we can say it was very hard. So we are going to discuss the questions. First we will going to discuss the questions from part B, and then we are going to discuss questions from the part C. I am discussing these questions according to my knowledge and expertise. Opinions may differ. The actual answers would be the answers which would be according to the CSIR answer key. So let's start discussion of the part B. So these are first few questions from biochemistry. These are the questions from series A of CSIR NET June examination 2019. So again, if you are having a series B or a series C, you can find these questions in your series, because the number may change. I am discussing the questions according to the series A of the NET examination. Question number 21. Question number 21 says that which one of the following statement on a protein conformations detailed below is incorrect? Such kind of questions it has been asked multiple times. So let's go through the options one by one. First statement says that L amino acids can occur in type 1' beta turns where phi and psi values are positive. Yes, this statement is true, because in class we have discussed that if it is a type 1 beta turn, it is type 1 beta turn, the amino acid which is at nth position, the phi values would be minus, you can see minus 30 and minus 60. Phi values is expected to be minus 90 and 0. But if it is a type 1' beta turn, then in this case the whole values become positive. The phi values for an amino acid which is at nth position, it would be plus 30, plus 60 for the psi. Similarly, the amino acid which is at the n+1 position, you will get a phi value of plus 90, and the psi value is approximately plus 0. So you can say phi psi values for a type 1 turn as minus 30 and minus 60, and the phi psi values of type 1 n+1 amino acid would be minus 90 and 0. But this time the question was asking about type 1 beta turn. So we can see that phi values, they are also positive. Psi values, they are positive, although psi value is almost 0, or it is slightly positive, it would be plus 1. So you can say that the option 1 is correct. Statement second says that a peptide rich in proline is unlikely to adopt a helical structure. Yes, we know that proline is a helix breaker, so this cannot be the correct answer. So this is a correct statement, that cannot be a correct answer, because definitely proline will not allow to adopt an alpha helical structure. The statement third says that proline residues have a high propensity to occur in a beta turn. Yes, this statement is true. We have already discussed in a class that often the amino acid which is at n+1 position, you will find a proline. At n+2 position, there could be glycine or some other amino acid. This is nth amino acid, hydrogen bonded with an n4th amino acid. This is the hydrogen bonding. So this statement is true that in beta turns, the proline is one of the amino acids which you can say that this is commonly occurring. Then moving to the question number 4th statement, the dihedral angle phi and psi of an amino acids in unfolded proteins are exclusively positive. No, this is false, because we know that psi and phi values for a random coil or unfolded protein, they lies in all 4 quadrants of a Ramachandran plots. That is, these could be positive as well as it could be negative. So the statement 4th is an incorrect statement. Question number 22, again from a biochemistry. This is question from the enzymes. This question is very easy. You have to choose an incorrect statement. The first statement says that the kinetic properties of allosteric enzymes do not diverge from Michaelis-Menten behavior. This is false statement, because we know that Michaelis-Menten enzymes, they behave a Michaelis-Menten, they obey a Michaelis-Menten equation. Their curve is rectangular hyperbolic, while the allosteric enzymes, they have different sort of kinetics. They obeys the Hill's equation. They shows a cooperativity, and we can say the curve is sigmoidal. So the first statement is incorrect. Rest of all statement, they are true, that in feedback inhibition the end product of pathway inhibit the enzyme of the pathway. That's true. Even the statement third is also true, that an antibody that binds tightly to an analog of the transit state intermediate of the reaction substrate to product would promote formation of product when the analog is added to the reaction. Yes, that's true, because we have studied in AB enzymes. This is the way how an AB enzymes they are prepared. That is, here we use an transition state analog of substrate as an antigen, and we obtain an antibody, and these antibodies they act as enzymes against a actual substrate. Statement fourth, the statement has been in options in CSIR net examination multiple times. The statement says that an enzyme with a kcat of 1.4 into 10 to the power 4 and km of 9 into 10 to the minus 5 molar has the activity close to the diffusion control rate. Yes, this statement is true. This is almost around the diffusion rate constant. So false statement is statement first. Now moving to question number 23. This is also a question from biochemistry, comparatively from a DNA sections, from a Chergaff rule. This is very straightforward, and we can say easiest question in a biochemistry which can be easily attempted. The questions says that the percentage of cytosine is 20%, then what would be the amount of adenine and thymine put together? According to Chergaff rule, which is applicable on our double-stranded DNA, we know that if cytosine is 20%, then according to Watson-Crick hydrogen bonding and Chergaff rule, guanosine would be also 20%. So cytosine, guanine both makes 40%. Rest of things would be adenine, thymine put together, they would be around 60%, out of which an adenine is expected to be 30% and thymine is also expected to be 30%. Here we were to estimate the amount of A and T put together, so answer would be 60%. Option 4 is correct. This is the easiest question from the biochemistry. Question number 24. This is also very easy question from biochemistry, metabolism, more accurately from a glycogen metabolism, from a glycogenolysis. The question says that the first step in a glycogen breakdown release glucose unit as. We know that the enzyme glycogen phosphorylase, it acts on our glycogen, and phosphorylase, as name suggests, they incorporate an inorganic phosphate during the breakdown, and glucose it is released in form of glucose 1-phosphate. So correct answer would be glucose 1-phosphate. Same glucose 1-phosphate then by the enzyme mutase is converted to glucose 6-phosphate, and then this glucose 6-phosphate could be further converted into a glucose by an enzyme glucose 6-phosphatase, and then glucose is released into the bloodstream from the liver. So immediate release of a glucose from a glycogen by the action of glycogen phosphorylase, it is in form of glucose 1-phosphate. So these were the 4 questions from biochemistry. Now we are discussing the questions from the cell biology. Next 4 questions, they are expected to be from the cell biology. This question is from sodium potassium pump, and we know that sodium potassium pump is a P type of pump. It has a phosphorylation domain where there is a conserved amino acid residue, that is aspartic acid, and on the R chain of aspartic acid there is a phosphorylation which induces a conformational change from E1 state to an E2 state of a transporter, and then sodium they are transported outside. So let's go through the question. The sodium potassium ATPase pumps from the plasma membrane, found on the plasma membrane of most of animal cells. Yes, a mutation in intrinsic phosphorylation site of pump would most likely. So we know that this is the you can say E1 state of this transporter. E1 state of the transporter. These could be alpha and these could be the beta subunits. The first step is that there is a binding of sodium ions. So sodium ion they binds over here. Once 3 sodium ion from inside they bind at this site, transporter domain, there is a binding of ATP on a nucleotide binding domain, and once an ATP bind, there is a ATPase activity. ATP is converted into an ADP and phosphate group attached to a phosphorylation domain, and once there is a phosphorylation, it induces a conformational change from E1 state to an E2 state, and in this condition, once it moves to an E2 state, 3 sodium they are released outside, and there is a binding of 2 potassium ion. and later on by phosphatase activity, phosphate group is removed and then potassium is taken inside. Now, if the site where there is a phosphorylation is mutated, the E1 state of the transporter will not convert to an E2 state. The result is that sodium ion, they will not be pumped outside, and if the transporter will not be in an E2 state, it will not have an affinity of potassium ion. As a result of this, even potassium ion, they cannot be imported into the cell. So correct answer would be that if there is a mutation on the intrinsic phosphorylation site of pump, it will affect both the movement of sodium ion outside and potassium ion inside. So third option is correct. Question number 26. This is the again question from the cell division part of the cell biology. Again a very straightforward question. The question says that the site of division plane during cytogenesis of animal cell is determined. You have studied in developmental biology also. Suppose this is the cell, and where a cell is going to divide, this depend upon the position of the spindle fiber. So this is suppose the spindle fiber. This is the spindle fiber, these are the chromosomes. The cell will divide exactly at the mid of the spindle fiber. This will divide like this, and it might happen that one of the cell is comparatively smaller and another cell could be larger. So plane from where a cell is going to divide, it is decided by the spindle apparatus. Almost at the center of spindle apparatus, there is a formation of contractile ring, and then there is a cytokinesis. So we can say that cytokinesis in case of animal, it is decided by the central spindle. The prophase bend, it may play some role in a plants. It do not have any kind of role in animal system. Randomly cannot be answered. Position of nucleus do not play any role in a cytokinesis. Question number 27. This is also from the cell biology, or you can say it has some connection with an animal biotechnology as well. It is also a topic of accessible matrix. Means this question which could be asked from multiple units. The question is that we know that in plant tissue cultures, in animal cell cultures, the primary culture is raised, and where the cells, they shows an anchorage dependent growth. There is a cell to cell and cell to cell. For raising a primary culture, first we need a cell suspension, and for obtaining a cell suspension, we have to break a cell to cell and cell to matrix interaction, and for this we utilize a variety of reagents and proteases. For example, the EDTA is utilized as a chelating agent. For example, it can chelate the calcium ions so that cell to cell interaction can be disrupted. Trypsin is a protease. It can break multiple proteins which are involved to cell or cell to matrix interaction. Collagenase could also be utilized because collagen is a major protein of an extracellular matrix. So these are three utilized. There is no role of a saprase in obtaining a cell suspension for raising a primary culture. Saprase, we know that it is one of the enzyme, or we can say one of the protein which plays a very important role in cell cycle at an anaphase stage, and we know that due to an activation of anaphase promoting complex, there is an activation of saprase, and this saprase basically somehow it leads to degradation of sister chromatid cohesin, and then the two chromatids of a chromosomes, they get separated. So saprase, it has a role in a cell cycle, especially at an anaphase stage. Then question number 28, basically this is a question from genome organization which is mentioned in unit 2. There are some statement, and again we have to identify the statement which is false. You can see that majority of the questions which are being asked in the CSIR exam nowadays, here you do not have to find out correct answer, you have to find out which is the incorrect answer. So obviously this is little bit difficult because you must be aware of 3 correct options, and then only you will able to weed out that which is not correct. So let's see. The physical, this is basically question from a nucleosomal organization of a core particle. In a core particle, we know that there is a hierarchical organization of a genome. First there is a nucleosome model, then there is a solenoid model, then there is scaffold model, then there is a loop domain, finally chromatid and chromosomes. These are different hierarchical levels. The first statement says that the physical structure of DNA is altered in middle of core particle. The physical structure of the DNA, it get altered in the middle of core particle, means somewhere here it get altered. Yes, that is true, because here almost DNA become flat. It become physically, it assume a different kind of conformation when it comes in contact with the proteins. When a minor groove of the DNA interact with the proteins, immediately carry out hydrogen bonding, the physical structures get altered. That statement is true. The second statement says that second statement is simply an extension of the first statement. In the core particle, DNA is organized as a flat superhelix. Here we are saying that the physical structure of the DNA get altered, and this is the extension of this point, that in the core particle, DNA is organized as a flat superhelix with 1.65 turns around the stone octamer. Yes, this statement is also true, and this statement is also true. Next statement says that while forming a 30 nm fiber, generally 6 nucleosomes per turn organize into 2 starch helix. Yes, that's we know. 30 nm fiber, earlier hypothesis was a solenoid hypothesis, according to which there is a formation of 1 starch helix. But now the presently accepted hypothesis is a zigzag model for a 30 nm fiber, and according to this, these 6 nucleosomes, they organize in a 2 starch helix. So this statement is also true. Statement 4 says that the N-terminal stone tails in a core particles are strictly ordered. No, this is false. They are disordered. The N-terminal tails of a stone tail in a core particle, they are strictly disordered. They no doubt radiate toward the extrinsic side. They are the site for various kind of modifications, post-transition modifications. They regulate the structure of chromatin. But this is false, that they are ordered. They are not ordered, they are disordered. So this is the false statement. So this is a false statement, and we have to find out the false statement. Moving to question number 29. Basically this is question from molecular biology. During replication, an RNA is H. RNA is H, you know, this is a nuclease which removes the RNA which is in hybrid state with the DNA, and due to this activity, basically it is able to remove an RNA primer. What is saying that this can remove an RNA primer. It can remove an RNA primer. So suppose this is the template strand, this is the RNA primer, and this is the DNA which is being extended. It can remove the RNA except that nucleotide which is linked with DNA. This is why this is so. The reason is stated that it can cleave a phosphodiester bonds which is between ribonucleotides. So the phosphodiester bond which is between the ribonucleotides, they can be cleaved, but a phosphodiester bond which is between the ribonucleotide and deoxyribonucleotide, it cannot be cleaved by an RNase H. For this, we need another enzyme, as we have discussed in a class, that is a FAN1 endonuclease, or we can say FLAP endonuclease, that is basically involved in that removing that terminal ribonucleotide. So correct answer is option second. Then moving to question number 30. Question number 30 says that in human cell line, a large fraction of double-stranded DNA breaks are repaired by non-homologous end joining NHEJ mechanism. An inhibitor of FLAP endonuclease will affect. We know that there are two different mechanisms of removing a double-strand break. One is referred to as homologous recombinational repair mechanism, and another is referred to as non-homologous end joining mechanism. In NHEJ mechanism, basically these are suppose double-strand break, and basically there is an enzyme FLAP endonuclease. It plays an important role in a gap trimming. So this is a direct question, that if you inhibit the activity of FLAP endonuclease, the gap trimming will not happen, and if gap trimming is not happening, then next stage ligase will not able to seal the DNA strand. So correct answer would be gap trimming. Other options is that it recruits the DNA dependent kinase. This is the role of Artemis, you know, in a NHEJ. DNA unwinding, it is role of helicase. Pairing of microhomology region, this is played by some protein RAC-A homologues in the higher animals. So correct answer is that if you inhibit activity of FLAP endonuclease, in this condition gap trimming will not happen. Moving to question number 31. Question number 31 says that this question it has been asked multiple times, but this time it is in a different flavor. The question is, sugar puckering in a double-stranded DNA nucleic acid is exclusively C2 endo in double-stranded DNA. From double-stranded DNA, we mean that we are talking about B form DNA. No, it is not C2. C2 is endo in a double-stranded DNA. Yes, it could be true for a B DNA, but we have to find out exclusively. If it is a A form of DNA, you know that even C3 could be endo. C3 endo is in for double-stranded DNA. No, this is also false, because we know that for a B form DNA, C2 is endo, while for a A form DNA, C3 is endo. C2 is endo in double-stranded DNA. No, we know that for double-stranded RNA, it represents or it resembles like a A form of DNA, and whenever there is a A form of DNA, you will find that C3 would be endo. So this is also false statement. These all are false statement. C3 is endo in hybrid duplex between DNA and RNA. Yes, we know that whenever there is a formation of a duplex between an RNA and DNA, RNA-DNA, whenever hybrid is formed, the structure resembles with an A form of DNA. And if there is an A form of DNA, there would C3 would be an endo. Yes, that's true. So correct statement is option 4. Now moving to the next question. Eukaryotic mRNA are modified to process a 5' cap structure. We know that yes, in eukaryotic mRNA there is a 5' cap structure, and then at 3' end there is a polyadylation, poly-A tail. Which one of the following is incorrect statement? We have to again find out the false statement related with the functions of 5' cap structure. The first statement says that it protects the mRNA from 5' to 3' exoribose nuclease activity. Yes, if cap is there, it will not allow a 5' exonuclease to function. So this is the 5' exonuclease activity would be inhibited, because we know that there is a special type of phosphorylated bond, 5' to 5' phosphorylated bond is present at a 5' cap, which cannot be cleaved by 5' 3' exonucleases. Second statement says that it facilitates splicing of nestin transcript. Yes, because we have already discussed in the molecular biology, only the 5' capped mRNA, they are spliced readily. So the mRNA, or we can say heterogeneous RNA, which are lacking 5' cap, the introns, they are not removed. Third statement says that it protects the transcript from degradation by RNA's third family enzyme. This is false, because RNA's third family of enzymes, they act on selectively on double-strand RNA, and we know that heterogeneous RNA, that is precursor of messenger RNA, is not a double-strand RNA. So this statement is false, and we have to find out the statement which is incorrect. Fourth statement says that it facilitates the attachment to 40S subunit of ribosome. Yes, this statement is also true, because we know that the small subunit of ribosome with variety of translation factor, it first bind to the 40S ribosome subunit, and then it scan up to a Kozak sequence. So this statement is also true, this is also true, this is also true. Third statement is false. So third is the correct answer. Question number 33. Basically this is a question from immunology, or you can say unit 4, immunology or microbiology. This question says that which one of the following does not belong to a human antimicrobial protein and peptides at epithelial surface forming an innate immunity. So epithelial surfaces, that could be our skin or some other epithelials which is covering our, lining our body, they are releasing various kind of proteins or peptide as an antimicrobial agent, and we have to find out which is not an antimicrobial agent. The option 1 is lactoferrin. Yes, we know that it has important role in innate immunity. Defensin also has a very important role in innate immunity. Even calprotectin also has a very important role in innate immunity. We have studied about these proteins in the microbiology or in an immunology topic. There is no role of vimentin in a defense. We know that vimentin is a type of probably intermediate, it is an intermediate filament, type 3 intermediate filament. It is a cytosolic protein. It is not secreted outside. It do not have any kind of role in defense. So which is not belonging to an antimicrobial protein, answer is vimentin. Question number 34, it is a direct question. That is, what would be the term which would be utilized for a death upon a detachment? And the direct term for this is anoikis. Anoikis is a death when some cell get detached from the rest of the cells, and if there is a cell death, such means if cells, they are anchoring with another cells, they keep on surviving. If they get detached and then they die, for this we use the term anarchist. You know the term of necroptosis, you know the extravasation, metastasis movement in a cancer cell. Extravasation is movement of some cell from blood capillary to outside. Necroptosis is apoptosis, or we can say there is a cell death due to some reason, and then there is a triggering of an apoptosis, that is referred to as necroptosis. Question number 35, this is, we can say, a question which is related to awareness. That is, if you are a science student, you must know some diseases which are prevalent in recent past. This question is, fruit bats are known to harbor spread several viruses that can infect other animals and humans, and we have studied our recent episodes, probably in a Kerala, where the fruit bats, they were transmitting a Nipah virus. Now the question is that which one of the following virus not reported to spread by a fruit bats? Now that is straightforward, because even if you don't know anything, you know that HIV virus, it is a blood-borne virus. It is when our infected blood, it comes in contact with another person or a blood of another person by any way, through a needle, syringe, blood transfusion, or some other activities, then there is a transmission of HIV. There is no role of fruit bats in a transmission of HIV. Fruit bats, they are able to transfer viral diseases like Ebola, Nipah, or even SARS. Then question number 36, this is again a very simple question, straightforward question from immunology branch. Question says that in a type 1 hypersensitivity mediated asthmatic response, which one of the following is thought to contribute significantly to prolonged bronchospasm and build-up of mucus in asthmatic? That is the direct question. We have studied it when we were discussing lipids in biochemistry, and we have studied that echinocytes are compounds of 20 carbon derived from prostaglandins, derived from the arachidonic acids, which include prostaglandins, prostacyclins, leukotrienes, and thrombaxanes, out of which the leukotriene, which is produced by the action of an enzyme lipoxygenase from arachidonic acid, and this leukotriene basically it is responsible for bronchospasm or for a bronchoconstriction. So correct answer would be leukotriene. Thrombaxane, it has some role in blood clotting. Transforming growth factor beta, it has some role in cell cycle control, growth, apoptosis, etc. Contrutine is, we can say, heteropolysaccharide, so they do not have any kind of role in bronchospasm. Leukotriene, which is a kind of echinocyte 20 carbon compound derived from arachidonic acid, leads to a bronchospasm. Question number 37 says that in a certain plant, the mechanism where timing of anther discence and stigma receptivity do not coincide, means it might happen that the anther release first, when anthers they are released, stigma is not mature enough that it can receive the pollen grains, and there may be fertilization. So it is one of the way to avoid self-pollinations. This mechanism where there is a different temporal development of male reproductive organ and female reproductive organ, this is referred to as dichogamy. So correct answer would be referred to as dichogamy. Question number 38, basically this is question from the developmental biology. The question is from Xenophus embryo. Xenophus embryo, beta-catenin plays an important role in dorsal ventral axis development. We know that beta-catenin, wherever it would be present, it would be responsible for our dorsal axis formation. The question is, what would you expect in endogenous glycogen synthase kinase GSK3? GSK3 is knocked out by dominant negative form of GSK3 in ventral cells of an early embryos. So first we must know what should happen is that on the dorsal side, there is some disheveled, or we can say there is some inhibitor of GSK3, and inhibitor of GSK3 is there. GSK3 is not able to inhibit the activity of beta-catenin, and as a result of this, beta-catenin would be able to determine the dorsal side. This is the we can say cascade. But if GSK3 it is not allowed to function, GSK3 if it is not allowed to function, in condition the beta-catenin would be there, and if beta-catenin would be there, even the ventral side will get determined into a dorsal side. So generally on the dorsal side. The inhibitor of GSK3 do not allow GSK3 to function as a result of beta-catenin accumulate and leads to dorsalization of an embryo. On ventral side, there is some kind of inhibitor of GSK3 which do not allow GSK3 to function. As a result of this, the GSK3 inhibit the beta-catenin. Beta-catenin is degraded in that side, get converted to you can say ventral side. But if GSK3 is not there, beta-catenin will accumulate even in on ventral side, and even that side which has to be a ventral also get converted to a dorsal side, or we can say a second axis will be formed on both sides, because from the dorsal side then there would be an invagination, gastrulation process would be initiated, and the second embryonic axis would be initiated. It would be something like this. Is this the axis from both the sides, gastrulation will start from ventral side as well as from dorsal side, or you can say a second axis will also be formed. Question number 39. This is a question from developmental biology. This is a very specific question. Basically the question is from homeotic genes. First let's go through a question and then we decide then we will decide which is the correct answer. Homeobox transcription factors, Hox proteins, play an important role in specifying whether a particular mesenchymal cell will become stylopod, zygopod or autopod in a lymph. Yes, we know. Basically, HOX-D genes, they play an important role. A model was proposed where these HOX genes specify identity of lymph region. Yes, a model we have already discussed that what is the role of HOX-D 1, 2, 3 and various other important HOX-D genes we have discussed. What would be the observed phenotype of a human homozygous for a HOX-D13? Now this is very specific. If you can't guess unless you know the function of HOX-D13 gene. Basically, HOX-D13 gene, it is involved in the morphogenesis of a limb, and in this case what will happen is that the flaps which is between the fingers, which should be removed due to an expression of HOX-D13 gene, they will not remove. This will remain fused. There would be an abnormalities of hands and the digits will fuse. Such answer would be the option 2. So there would be no, you can say, change in stylopod or a zygopod. There would be a changes in an autopod. Question number 40. Basically now this is question from mineral nutrients, question from plant physiology, mineral nutrients. The question is, what is the function of silicon in plants? And we know that in grasses they are quite abundant in silicon. They need the silicon and is one of the important nutrients for the plant, for the grasses. What is the role of these silicons? First students, it is consisted of amino acids. We know that standard amino acids except silicosis, means almost all 20 amino acids, they do not have silicon, they do not have any kind of role in amino acids. Second statement, this says that contribute to cell wall rigidity and elasticity. Yes, that's true. Grasses especially, they have a rigid cell wall and the elasticity is also due to the silicon. Constitute the photosynthesis reaction center. We know that this role of manganese, maintenance of cell turgor and electroneutrality. Here major role is played by potassium and some other ions. So correct answer would be the option 2nd. Now question is that most of the plant resistance genes which code are receptors, and then there are some cytosolic domain. We know that the region of the receptors which interact with a AVR gene, basically it has a leucine-rich repeats, and on cytosolic side there is a nucleotide binding domain. So correct is, this is straightforward question from plant pathogen interaction, and this question is that the plant disease resistant gene products, that is R gene products, they contain a leucine-rich repeats. So option 3rd is correct. Question number 42. Out of several gibberellin identified in plants, which one of the following is not bioactive? Again, this is question of cramming. You have to have a core information about the active form of gibberellic acids. The active gibberellin include GA1, GA3, GA4, GA7. These are some of the active form of gibberellic acid, which has a hydroxyl position, OH position at beta or we can say they have generally beta 3 OH groups, they have a carboxylic group. These are some of the important components of an active form gibberellic acid. Especially GA5 and one more GA6, these are two gibberellin which are lacking OH group at beta 3 beta 3rd position. They are lacking a hydroxyl group, and under these conditions they remain in the inactive form. So GA5 is not bioactive because it is lacking a hydroxyl group at a beta third position. Question number 43 says that nitrogen is a complex metal containing enzyme is involved in conversion of nitrogen to ammonia. Yes, that's true. Which of the following metal is not involved in the activity of nitrogenase? We know that nitrogenase, which is composed of two different subunits, it needs ferrous as one of the component, it needs molybdenum as a cofactor, and there are some forms of nitrogenase which also need vanadium. Now the question is, which one of the following metal is not involved in the activity of nitrogenase? So molybdenum is involved, iron is involved, and even vanadium is involved. So correct answer is cobalt. There is no role of cobalt. So correct answer is not vanadium. The correct answer is cobalt. There is no role of cobalt in a nitrogenase. It needs majority of nitrogenase which occurs from in the bacteria. They need iron, they need molybdenum, but there are some forms of nitrogenase which may utilize vanadium as well. So which is not being utilized is the cobalt. Moving to the question number 44. These are the now questions from animal physiology. Which one of the following agent cause relaxation of mesenchymal cells? Basically this question is from the excretory system, this is from the kidney, and basically it is from the regulatory part, that how a glomerulus filtration rate is being regulated. We know that just beneath the glomerulus epithelial cells there are some specialized connective tissue cells referred to as mesenchymal cells, and these mesenchymal cells which could be excited by histamine or norepinephrine. The question is not about the excitation, the question is about the relaxation. They are relaxed by dopamine. So this statement is true. This is a straightforward question, that mesenchymal cells of a kidney which are involved in glomerulus filtration rate, they are relaxed by the activity of dopamine. Question number 45, which is again from animal physiology. Basically this question is from the nutrition part. Question is, a patient comes to hospital complaining of vomiting and diarrhea. Okay, the doctor suggested that patient take glucose and electrolyte solutions. Means basically you want to say that glucose should be taken with an electrolytes like sodium. Which one of the following membrane protein is likely to be involved in rehydrating the patients? That is a straightforward question. We know that on the active phase of epithelial cells there is a sodium potassium pump. So glucose sodium glucose pump. So glucose would be actively uptaken if only there is a gradient of a sodium ion. So rehydration would be much better if he or she is taking an electrolyte, because sodium gradient will be utilized for the active transport of glucose, and the transporter which is involved is referred to as sodium glucose transporter protein 1 or SGLT. Cystic fibrosis transmembrane conductor regulator, you know that it is involved with transport of chloride. Insulin receptor protein, it doesn't have any role in glucose transport, it is involved in insulin signaling. Sucrose isomaltase protein, basically it is involved in handling the sucrose and isomaltose. So answer would be 2. Question 46, it is again a straightforward question, that which one of the protein is basically required for vitamin B12, that is cobaltamine absorption in a small intestine. This we have already discussed in a class, probably Pransu sir would have discussed, I know he have discussed this in class, that vitamin B12, it is uptaken by the cells of ileum of small intestine by receptor mediated endocytosis, and for this we need some kind of intrinsic factor referred to as cobalamin, and this cobalamin, it is released in a saliva, and in a stomach in an acidic environment there are various proteins, and vitamin B12, when it is released from the protein, this vitamin B12 it binds to a cobalamin in the acidic environment in the stomach, and then this cobalamin vitamin B12 complex, it migrates to the small intestine, especially in the ileum part, and then there are receptors for cobalamin, there is a receptor mediated endocytosis, and then there is a uptake of vitamin B12. So the answer would be that protein which is required for vitamin B12 absorption in small intestine is a cobalamin. Then moving to our question number 47. Basically this question from again animal physiology, from sensory organs, from the eyes, that vision, that how light it interact with the rod cells, and then there is a signaling. We know, we have already discussed that these are the rod cells, and within the rod cells these are the disc-like structures on disc-like structures. You can find these are the rod cells, disc-like structures, and on the disc-like structures there are some photoreceptors referred to as rhodopsin. And in the dark, the sodium channels, they are open, sodium ion, they move inside. There is a depolarization of a membrane, and the rod cells, they start keep on releasing the neurotransmitter like glutamate. Now if there is a interaction of light, if light interact on the rhodopsin, there is a conformational change in an opsin protein. Once there is a structural change in rhodopsin protein, it activates a associated G protein referred to as transducin. Once a transducin is activated, transducin activates a phosphodiesterase. Once a phosphodiesterase is activated, it converts a cyclic GMP into a GMP, so the concentration of cyclic GMP decreases. The ligand-gated sodium channels, especially cyclic GMP-gated sodium channels, they get closed. The membrane which was in a depolar state, it get repolarized, and once it get repolarized, there is a now lesser release of neurotransmitter glutamate. So correct answer would be the sequence of events are mentioned in option 3rd. Now moving to the next question, question number 48. This is now question from a genetics part. LOD score of 3 represents a recombination frequency, that is LOD is a logarithm of odd, and we know that LOD value is 3, so anti-log of 3 would be 1000, and this simply suggests that the data, they are in favor of a linkage 1000 times more as compared to the linkage, as compared to a independent assortment. So the LOD score of 3, it suggests that recombination frequency is 100 times as likely as an hypothesis of no linkage. So this would be the correct answer. Question number 49, basically again it is question from genetics, little bit mathematics is involved in this question. Assume that A, B, C, D genes are not linked, means there is an independent assortment. The probability of progeny being this, what we need is capital A, capital B, small c, capital D, small d. This is needed. From a cross between one of the parent is capital A, capital B, small c, capital D, small d, and another parent is small a, capital B, small c, and capital D. If they are crossed, we have to obtain this kind of genotype. What is the probability of getting this kind of genotype? So we can, as we have already discussed in a class, this could be easily done by the fact that if there is a cross between capital A and small a, 100% you are going to get capital A, small a. That is sure. A cross between capital A and small a, definitely it is going to give you a capital A, small a. Second is a cross between capital B, small b, and capital B, small b. Capital B, and a cross between capital B, small b. It can produce capital B, or it can produce capital B, small b. 50% chance is this, and 50% chance is that. So the chance of capital B would be half. Then the cross between small c with a small c, and chance that you will get small c is 100%. Again, this is the only chance. What is going to happen? Again, if you cross between capital D, small d with a DD, only two choices are possible. Again, you can get a DD or you can get a DD. The chance of getting this is half, chance of getting this is half, and this is being asked. So chance of this getting is the half. Chance of getting capital A, small a, capital B, capital C, capital D, small d is multiplication of this, and this would be 1 by 4. So correct answer would be 1 by 4. Moving to question number 50. Question number 50 says that a newborn baby of a mother having a blood group AB. So the genotype of a mother is sure, it is IAIB and RH positive. So we can write this RH positive dash. Means we cannot say that she is heterozygous or homozygous. And father is having a blood group O. Father is having blood group O. So genotype has to be IO, and RH negative means surely that individual would be Rh negative, homozygous for blood group allele as well as homozygous for the Rh factor. Now the baby which of the following group is expected to be Z-double. So expected could be either a blood group A, or expected could be a blood group B. These are the two possibilities. The blood group could be A or a blood group could be B. From Rh point of view, it could be Rh positive or even it could be Rh negative. So first statement says O is not possible, O is not possible, AB is also not possible, only possibility is B. Yes, this is possible, and Rh positive is possible. So correct statement would be option 4. Question number 51. This is question from the markers. Such question it has been asked multiple times. Which one of the following DNA marker can be used to distinguish between homozygous and heterozygous? Basically, this is asking about a codominant marker. And we know that some important codominant markers include RFLP, or it include SSR, or it include STR, SSR, STR, VNTR, etc. These are some of the example of codominant marker, and even SNP is also a codominant marker. So the question, and these codominant markers, they are able to distinguish between homozygote and heterozygotes. RAPD, RFLP and ISSR, these markers, these are dominant markers. They cannot distinguish homozygote and heterozygotes. Only out of these choices, RFLP is the only codominant marker. Moving to question number 52. Which of the following is a fungal disease of the plant? Basically, this is now question from the diversity part, where some questions they are being asked from animal disease or plant disease. First question is cucumber mosaic. We know that cucumber mosaic disease is caused by cucumber mosaic virus, so that cannot be answered because it is not a fungal disease. Fire blight of pear. The fire blight of pear, basically it is a bacterial disease caused probably from a Irvinia species, so this is also false. Crown gall of various dicots. This is straightforward, caused by bacteria, especially Agrobacterium species, Agrobacterium tumefaciens, so this cannot be an answer. So the fungal disease out of this is apple scab. Apple scab is a disease of Melis plants. Basically, it is caused by Ascomycetes fungus, and the name of that fungus you can note down is Venturia iniculus. This is the fungus of Ascomycetes, basically, which leads to an apple scab. Then question number 53. This is again a question of general awareness. The question is that which one of the following influenza A virus subtype caused severe avian flu and was responsible for disease outbreak in year 1997 in Hong Kong? So the important thing is that you have to first focus on avian flu, and it was responsible for 1997 Hong Kong. This is a very typical question, unless until you are aware you cannot take a risk. So correct answer is an H5N1. This is the one of the avian flu virus, and it was responsible for causing a disease in basically in the chickens, in the avians, birds in Hong Kong. H1N1, it is major influenza virus which causes disease in human. It is one of, especially in 2009 there was an outbreak of H1N1, and the disease was being caused from a person, human to human. H797 is comparatively having a low virulence strain. It can also cause a disease in human, pig, and various other species like horse, etc. Similarly, H3N2 can also cause a disease in a human, pig, and some other species, but they are not responsible for avian flu. Question number 54. Basically, this question is from phylogenetic tree. This is a simple, straightforward question. Basically, here you must know what you mean by monophyletic and paraphyletic. So if you are having one common node, and from common node you are including all the species which are derived from this, for example species B, species C, then such an taxon is referred to as monophyletic. You are having one common ancestor, but you do not include all the species. Suppose you are making a grouping like this where you are including A, B and C but not including D. Such kind of taxons are referred to as paraphyletic. And if it happens that the species which do not share any kind of common ancestry and they are put together, such an taxons are referred to as polyphyletic. So in group X you can see that it include A, B and C. They have a common ancestor, and all the lineages which derived from common ancestors, they have been included in this grouping. So such an taxon would be referred to as monophyletic. In case of group Y, you can say that in group Y, D, E and F, they have been kept. They also share a common ancestor. The lineages which derived from common ancestors include D, E, F and G, but all the lineages they have not been included. G has been excluded, and such a kind of taxon would be referred to as paraphyletic taxon. So group X is monophyletic, while group Y is a paraphyletic. Moving to question number 55. Basically this is question what you would have studied somewhere in a BSc classes. This was a very simple, straightforward questions used to be asked in our viva examinations during a BSc practical examination, that what is the differences between a poisonous snake and non-poisonous snake. So if you remember something, that the characteristic of a non-poisonous snake is that they have a cylindrical tail, while in comparison to cylindrical tail and small belly scales, while comparative to this the poisonous snakes, they may also have a cylindrical tail, but they have a broad transverse belly scale, and fourth infralabial scale is the largest, while the poisonous snake, they could also have a flat tail where a broad transverse scales, and with the broad transverse scales, small belly scales, they are present only in non-poisonous snakes, while the third supralabial touches eye and nose. Yes, that is also true. They could also have a cylindrical tail, but poisonous snakes, they have a broad transverse belly scales. Only the thing is that if you are in touch with this topic, that non-poisonous snakes, they have small belly scales. So on basis of this, you can move closer to the answers. Correct answer will be option 1. Question number 56. Basically now these are the questions from we can say diversity or ecology part, species richness. From that, this question has been asked. Question has been asked that a road is constructed through a tropical forest. So there was some kind of, you can say, tropical forest, and there were the butterflies everywhere, and a road has been constructed. Suppose this is the road which has been constructed in this tropical forest. A road has been constructed in a tropical forest, and it has been assumed that with this, the species of forest butterfly could decline. Some species could decline. Which of the following is not a possible explanation for road causing a decline in the forest butterfly population? So which is not a reason. First is that road facilitate immigration of gap-loving species which compete with the forest species. Yes, that's true, because wherever there would be a road, there would be a gap between the forest, there would be a patch, and there may be some gap-loving species. They can utilize that niche comparatively better and could outcompete these native butterflies. So this could be a correct answer. Second statement says that road facilitate increased movement of forest butterfly within the forest. No, it is not possible, because it will act as a barrier. It will not allow easy movement. And even if you assume that if it allow increased movement, it must increase the genetic diversity. It says that it is reducing genetic diversity. No. Genetic diversity get reduced when the species get remain localized or endemic to some of the part. So this statement cannot be a correct explanation why the species will become extinct, because if there is a rapid movement, it should increase the genetic diversity. It will allow a mixing of gene pool. Statement third says that road internally fragment the habitat and negatively affect important microhabitat condition for the first butterfly. Obviously, first it is fragmenting the habitat into two parts, and the habitat which is around the road, that is the microhabitat, they will get also disturbed, and due to which various species they may become extinct. That is also true. Fourth statement says that road facilitates invasion of non-native plants that displace native host and nectar plant from the first butterfly. Yes, that's true, because you are creating a new kind of ecological habitat, and that may be more favorable to certain invasive plant species. Some different kind of plant species may be there instead of the native plant species, and the native plant species which were harboring a butterfly, which were supporting a butterfly, providing nectar to butterfly, if they are not there, the butterfly species dependent on such kind of plants, they will also become extinct. So this is also a correct explanation. So the incorrect explanation is option second. Moving to question number 57. Which one of the following statement regarding normal distribution is not correct? Normal distribution, as you all know, it shows a bell-shaped curve. There is a bell-shaped curve for a normal distribution, and there is a symmetrical, you can say, distribution around mean, median and mode. So the question is that which is not correct. So it is symmetric around the variance. No, it is not symmetric around the variance, it is symmetric around the mean, median and mode. So statement third is not correct. Now moving to question number 58. Again the question you are asked, this question is being asked saying that which is not true, means again you are going to an exception. Tropical regions have more species diversity. No doubt tropical regions, they have a more species diversity, and there are some possible explanations that why the tropical species they have more diversity. The first explanation which is given is that tropical regions had a more limit to diversify under relatively stable climatic conditions than temperate regions. Yes, that's true. Second statement says that tropical regions have high spatial heterogeneity. That is also true. These statements we have already discussed in our class. Third statement says that greater biological competition in a tropic leads to a narrow niche, and if niche is narrow, more species they can remain in that area. Yes, that is also true. But you can look at the fourth statement, which is totally false. It says that lower predation intensity in a tropic allows survival of more prey species. No, if you remove a predator or if you remove the predator pressure, there would be dominance in few of the prey species, diversity will get reduced. That we have discussed in a class. So that's why predators, they are often a keystone species because they maintain the complete structure of the forest. So if the predator intensity would be lesser, there would be more increase in the population of prey species. Prey species, they will start competing with each other. One of the dominant prey species would outcompete the, we can say, another inferior prey species, due to which the species diversity can decrease. So the statement fourth is not a correct explanation. Question number 59. This is a direct question between association of fungus and plant. That is basically question is from mycorrhizal. And we have to identify that which statement is correct statement. The first statement says that it states that ecto-mycorrhizal association predominantly reduce phosphorus limitation. No, ecto-mycorrhizal association, which form a Hartig net, or you can say some another structure, mantle-like structure around the cells, it reduce both phosphorus as well as nitrogen. And endo-mycorrhizal association reduce both nitrogen. No, endo-mycorrhizal reduce only the nitrogen limitation, which form a vesicular or vesicular-like structures. So statement second says that endo-mycorrhizal association predominantly reduce phosphorus limitation. Yes, that's true. And ecto-mycorrhizal association reduce both nitrogen and phosphorus limitation. Yes, this is also true. The correct answer would be statement 2. Moving to question number 60. Which one of the following statement is correct for the process of speciation? And we know that speciation, it may occur by multiple processes. Allopathic speciation, where geographical barrier is important. Then there is a sympatric speciation, where within a same place there is a formation of new species, probably due to hybridization, polyploidy, and some other factors. And paraphatric speciation, where the speciation occurs at adjacent places, basically due to variations in an environmental gradient. First statement says that allopathic speciation occurs between adjacent populations. No, it's not allopathic speciation here. If it would have been parapathic, then it would have been okay. In allopathic, there are two different places, they are not adjacent places. Parapathic speciation may occur between geographically separated population. No, this is true for allopathic speciation. This is also a false statement. Third statement says that sympatric speciation occurs within one continuously distributed population. Yes, at the same place, in one continuously distributed population, at the same place, the speciation which occur is referred to as sympatric speciation. This statement is true. Fourth statement says that sympatric speciation occurs when continuously distributed populations are fragmented. No, this is false. If there are fragmentation, or there is some kind of, you can say, geographical barrier, under such conditions there could be a peripatric type of speciation, and if this geographic barrier is large enough which isolates the two populations apart, then there could be in this condition there could be allopathic speciation. So this is also false. So correct statement is statement third. Question number 61. Which one of the following does not contribute to microevolutionary changes? All those factors which brings a change in allele frequency and thus in a change in genotype frequency are responsible for microevolutionary change. Mutations bring changes in allele frequency, we know. Genetic drift makes an abrupt change in the allele frequency. Natural selection brings a change in an allele frequency according to the habitat and adaptations that we already discussed. Random mating is the only phenomenon which do not bring any kind of change in allele frequency and genotype frequency, as we have discussed in Hardy-Weinberg equilibrium. If it would have been a differential mating or assortative mating, there would have been some change, but random mating do not bring any kind of change in allele frequency, or it is not responsible for a microevolutionary change. Question number 62, basically this is from animal behavior part. Multiple time question has been asked from a Hamilton rule, which is a rule related with kin selections, and this time from this Hamilton rule which explains the allotic behavior for close relatives. This question is very straightforward, that according to Hamilton rule, the allotic act will occur when a genetic readiness and benefits they are greater than the cost. Where B is benefit, C is cost. What we mean by R? Genetic readiness. So genetic readiness should be between the performer who is going to carry out a allotic act and the recipient for whom this act is being done. So we have discussed that the genetic readiness between the performer and his own son or father or daughter is 0.5. The allotic, the genetic readiness between this is for father, son, brother, etc. Similarly, the genetic readiness for a grandson or grandfather is 0.25. We have already discussed in a class. So this is R is a genetic readiness. The genetic readiness, who is going to sacrifice, who is going to perform, and the genetic distance between the recipient for whom this allotic act is being done. So statement 2 is correct. Question number 63, again a straightforward question, that analysis of nucleotide sequence of ribosomal RNA are particularly useful for evolutionary studies of living organisms. We know that rRNA it is utilized for phylogenetic studies, and there is a host classification according which due to which we were able to distribute all the organisms to 3 different groups, kingdoms, like eubacteria or archaea or eukaryota. But why this rRNA is being utilized? So there are multiple reasons, and we have to reason which is not true. Means we have to find an false statement. First statement that rRNA is evolutionary ancient. Yes, it is evolutionary ancient. It is present in the archaea, it is present in eukaryota, and it is present in the eubacteria as well. No free living organisms lack rRNA. That is also true. All free living organisms, they have to produce a protein, and for proteins they need ribosomes, and for ribosomes the important we can say component of ribosome is rRNA. This also statement is true. rRNA, since critical for translation, yes, that's true, can undergo lateral transfer. No, it doesn't get a lateral transfer. There is always a vertical transfer. Any DNA which is laterally transferred cannot be utilized for phylogenetic studies. The DNA which is transmitted from father to son to grandson in a lineages, only such kind of DNA or we can say molecular marker could be utilized for a molecular phylogeneity. If there is lateral transfer, now it has no role in a phylogeny. So third statement is false, and we have to find out the exception. Fourth statement says that rRNA has evolved slowly over geological time. Yes, that's true. rRNA is playing a very critical role in a protein synthesis, and any kind of changes in rRNA is accepted to very lesser extent. So if you compare an rRNA across the different kingdoms, you will find a comparatively very lesser change. This statement is also true. Question number 64 says that which one of the following is most appropriate definition of gene pyramiding in a plant? This is again direct question from a transgenic part of a plant biotechnology. Gene pyramiding means transferring multiple genes at a same time to a target plant species to get a resistance against some kind of stress or resistance against some kind of pathogen or some other goal. So correct answer is gene pyramiding approach is introducing different genes for resistance to a single pest in a single genotype. This is the correct answer. Question number 65. Basically this question is from the FRET. To test that cyclic AMP, there is some cyclic AMP binds to a protein kinase A. In absence of cyclic AMP, no FRET signal was detected. However, upon cyclic AMP addition, a strong emission of 530 amp was obtained. Now, which should be the proper combination of fluorochromes? Again, this question we have probably discussed in a molecular biology when we were discussing a FRET approach, that two fluorochrome which are routinely utilized, one of the protein or a molecule is tagged with a CFP. The advantage of using a CFP is that its excitation wavelength is around 430 nm and its emission wavelength is 480 nm. But if this protein it is interacting with another protein which is fused suppose with another protein referred to as YFP, the excitation wavelength for a YFP is 480 nm and the emission wavelength would be 530 to 535 nm, which lies in a yellow region. So two important protein which could be utilized for an emission of a yellow light would be a combination of CFP and YFP, because CFP is having an excitation wavelength of 430, emission at 480 nm. The same wavelength 480 nm is, and we can say excitation wavelength for YFP, and YFP will start emitting the wavelength around 530 or 535 nm. So correct combination would be option second. Moving to question number 66. Again we have to find the statement which is not true, means incorrect statement को हमें discuss करना है. In bioremediation by microorganisms detailed below, choose an incorrect options. First option says that organic contaminants provide a source of carbon. Yes, it provides a source of carbon. Organic compounds, they can provide a carbon. The bacteria do not get energy by degrading contaminants. If it's not getting any energy, then why it is degrading the contaminants? Definitely there must be some kind of energetic benefit to a bacteria, so this statement is totally false. It derives an energy on hydrolyzing or on breaking the bonds which is present in the contaminants. C statement says that bacteria can produce oxidized and reduced species that can cause metal to precipitate. Yes, that is also true. We have studied it in bioremediation. Bacteria act on contaminants by aerobic as well as anaerobic respiration. Yes, that's also possible. Aerobic bioremediation is possible. Anaerobic bioremediation is also possible. We have already discussed in class. Question number 67 says that a multimeric protein when run on SDS shows two bands of 20 kilodalton and another band is of 40 kilodalton. So these are the two bands. It means that's true that at least there are two type of subunit. One is of 20 and one of 40. So it cannot be homo. It must be a hetero. Now among the given choices, the total molecular weight is of, in native gel, the total molecular weight is of 120 kilodaltons. So if it is homodimer, total molecular weight would be 60 kilodaltons. So it is not 60 kilodalton, it is 120 kilodalton. So this cannot be answer. Heterotrimer, it cannot be, because these are two possibility, 20 20 40, or it could be 20 40 or 40 if we consider it as a heterotrimer. If it is the case, it would be 80 kilodalton. If this is the case, it will be 100 kilodalton. Again, this cannot be answered. Only possibility is that it is a heterotetramer, where there are two subunits of 20 kilodalton and there are two subunits of 40 kilodalton. So total molecular weight of this, that is, would be 120 kilodalton. So correct answer would be heterotetramer. This is the only combinations which is possible among the given choices. There could be one more option which is not given in the there could be some other alternatives as well. It could be like 20 20 20 20 and 40. It could have been like this also. So this could have been 80 and this could be 40, but this is not given in the option. So why we are moving in that direction. So correct answer would be a heterotetramer. Question number 68, basically it is from the spectroscopy part, basically from a Lambert-Beer rule. A solution contain NADH and NAD both at 0.1 millimolar concentration. Means it is having NADH and NAD. There are two things, NADH and there is an NAD. Concentration of this is given to you is 0.1 into 10 to minus 3 molar. Concentration of this is also given to you is 0.1 into 10 to minus 3 molar. That is 1 millimolar. 1 millimolar. This is the concentration which is given. The molar exchange coefficient value is also given. For NADH it is 6220, and for this molar coefficient is almost 0, you can say. The path length is also given. The path length is given is 5 millimeter, or you can say 0.5 centimeter. The path length is also 0.5 centimeter. We know that the total absorption would be the combination of ECl from NADH plus ECl from NAD, but since the molar exchange coefficient of NAD is 0, so this value will become 0. It would depend only on the concentration of NADH. So correct answer would be 6220 multiplied by concentration is given to is 0.1 into 10 to minus 3. Path length is given as 0.5. So you will solve this, you will get 0.31. So correct answer would be 0.31. Then the next question is question number 69. Orientation of clone DNA fragment in a plasmid vector can be checked by. So these could be the two different orientations. Suppose this could be orientation A and this could be orientation B. So in a plasmid vector. So how you can identify that the orientation is like A or orientation is like B? So what we can do is we can use one kind of primer which is specific for the gene fragment. Suppose this is the primer which is specific for a gene fragment and binding somewhere here after 1 kb. This primer is binding over here. Okay, suppose it is binding here. And another primer, suppose it is specific for vector and is binding somewhere here. One is acting as a forward primer and another is acting as a reverse primer. The result is that the DNA which is amplified, this region will get amplified if there is a correct orientation. If the DNA is reverse orientation, this one of the primer will bind over here at this region. Another primer, if it is binding at the same place, suppose this is binding at the same place, then the DNA which is flanking in between this will get amplified. So the DNA which would be amplified would be of different length, and you can easily distinguish whether it is an orientation A or an orientation B. So you must have one primer specific for the insert, another primer specific for the vector, and you can now distinguish the orientation of clone DNA fragments. So correct answer is option 3rd. Moving to the last question of part B. This is the last question which we are going to discuss. This is the straightforward question. The question is that emission maximum of tryptophan fluorescence in a protein is 335 nm. This suggests that tryptophan. We have already discussed that basically that emissions of a tryptophan is around 350 nanometer, but if most of time tryptophan being a non-polar amino acid, it is often seen at the interior of the protein. Here you can find somewhere a tryptophan, and here the environment is non-polar. And as discussed in a class, when the tryptophan is in a non-polar environment, there is a blue shift, and due to a blue shift, the emission spectrum, which is being, if you carry out an excitation, the emission spectrum comes out to be a 335 nm. So this is that question, that when our tryptophan, and mostly that happens, the tryptophan is in hydrophobic environment, the emission spectrum is around 335 nm. So these were few questions these were the questions from our part B. In next session, we are going to discuss question from part C. I have tried my best to solve these questions. There could be variations in the answer key. I am not taking any kind of surety, any kind of doubt or discussion is welcome. If you feel that some answer is wrong, you can discuss that, but this is the explanation according to me. So thank you for watching, and hope, especially for IfAS, you would have done most of the questions correct. So thank you.